Hello, welcome back. This is Professor Young, and this is our look at brain anatomy and the organization of your nervous system. This will basically serve as our final WIMBA together for AMP1. And so if you are listening to this, you have survived, and congratulations, you are almost there. There is a light at the end of the tunnel. And so with that said, let's jump into this and see where we go. And so before us here is a breakdown of the entire nervous system and the various divisions or chunks that we can look at and study. Of course, we know that when it comes to the central nervous system, what we are really referring to here is the brain and spinal cord. And when we refer to the peripheral nervous system, we are basically referring to everything outside of the central nervous system. All right. Now, notice that we can take each of these sections and kind of divide them down into smaller and smaller specificities, if you would. And so that's exactly what we're going to do. We can take the peripheral nervous system and break it down into two different neural components or functions. The sensory, which is going to basically send information. The job of that sensory there is to gather info and send it to the central nervous system. And so the sensory nervous system, with the sensory component of the peripheral nervous system, is going to gather information from the external world, or the external tissue, if you would, and send that information to the central nervous system. Whereas the motor function is going to carry out response from the central nervous system. So the motor function or the motor division of the peripheral nervous system will receive instructions from the central nervous system and respond appropriately. When we talk about coordination of motor functions, we are specifically dealing with skeletal, smooth, and cardiac muscle. That's what's going to respond based off of a motor function. So your skeletal muscle is going to respond in some way, your smooth muscle is going to respond in some way, or your cardiac muscle is going to respond in some way. Now, the motor function, as I've kind of already hinted at, can be divided up into how it responds. If the skeletal muscle is responding, then it is said to be a somatic response. If it is what we refer to as an autonomic response, then it is smooth. I can't spell smooth. And cardiac. It is smooth and cardiac that is responding. Autonomic Auto meaning self. Right? You don't have control over this. This is a voluntary response. If your blood pressure is dropping, then what will happen is vasoconstriction happens, which means your arteries become narrower, and very often cardiac output will increase. Well, that's not a somatic response. That's an autonomic response because you don't have to think about that. Your body's not going in and saying, "Oh, geez, blood pressure seems to be dropping. I wonder what I should do." You don't have to go through and do any of that. Boom, blood pressure drops. All right, your carotid pressure points within your neck, pressure receptors, baroreceptors, instantaneously sends a message to the medulla oblongata, where the medullary control center, cardiac control center, kicks into high gear and says, "Oh, blood pressure's dropping. Let's send some epinephrine down to the heart and to the blood vessels and cause vasoconstriction and increase that cardiac output to be able to pump up and bolster, if you would, that blood pressure." That's all. That's all an automatic response. Automatically happens, and so that's your difference between autonomic and sonomic. Right? Whereas the somatic is going to go ahead and you drop a pencil and you need to bend over and pick up that pencil. Well, you've got control over that. Right? You consciously have to sit here and say, "Oh, shoot, drop the pencil." Got to bend over, so now your lower back muscles have to contract, and your ab muscles have to contract, and your leg muscles have to be contracted a little bit, and then you got to go ahead and bend down, and then your muscles within your forearm and your hand have to contract. That's all stuff you have control of. And finally, we have the sympathetic and the parasympathetic nervous system, and I will say that in A&P2, this is exactly where you guys pick up with, and that's with the autonomic nervous system. Your sympathetic nervous system directly regulates smoothing cardiac muscle. All right. And it regulates it in such a way that it excites or increases activity. Right, so it's going to excite and increase activity within the smooth and the cardiac muscle. Parasympathetic, on the other hand, does something that we refer to as, or the term that I use is down regulates. It down regulates. It calms smooth and cardiac muscle. And so that's kind of your major overall divisions that we have within the nervous system. So again, the nervous system, two big branches are the central nervous system and the peripheral nervous system. Within the peripheral nervous system, you can also have sensory and motor functions. Within the motor functions, you can have either a somatic or an autonomic response. And within the autonomic response, you can either that response can either be an increase or it can be a decrease within the activity found within the smooth and the cardiac muscle. So this is a nice little flow chart here for you. Now, what we need to turn our attention to is a little bit more of some detail with the central nervous system. And that is where we are going next. And so the remainder of our presentation here will be all spent on looking at the parts of the brain. And the job here is to really break down the brain into basic regions, if you would, right? Basic regions. And those regions, those key regions for us are going to be the brainstem. It's going to be diencephalon. It's going to be the cerebrum. And finally, or maybe not finally, but within that area, the cerebellum. And so they're kind of the four distinct regions of the brain, anatomy and structure and function that we really need to look at. But before we get there, let's start outside of the brain first. And the outside of the brain, we know that we have this tri-layer, if you would, of protection that is referred to as the meninges. And the meninges, again, is this series of three layers that serves as protection, it also provides nutrients. It also removes waste. And so it's got three big functions here: protection, nutrients provisions, and waste removal. Those three layers are referred to as the dura mater, the arachnoid layer, and the pia mater. And on the next slide, we're going to specifically look at where each of these three areas are located. For right now, understand and remember back when we did the brain dissection, the dura mater is that milky white, leathery, tough exterior layer of the meninges. And what it does is it directly anchors the brain to the cranial cavity. And so in that sense, it's preventing the brain from really bouncing around too much within the cranial cavity, providing protection for the brain. The arachnoid layer is the innermost, is the middle layer, and it has a space within it known as the arachnoid space. And it's within this arachnoid space of the arachnoid layer, or the subarachnoid space, where we find a high concentration of blood vessels. It is where we find a very clear and sterile fluid known as the cerebral spinal fluid. And that cerebral spinal fluid is what is charged with the duties of delivering nutrients, delivering oxygen, and removing waste, as well as bathing the entire brain as well as the spinal cord. And when we use that term bathing, it's providing cushioning. You've got this liquid now that is providing cushioning to the central nervous system. And so it's providing both the role of nutrient supplier, waste remover, and protector. And then we have the pia mater, which is the deepest layer. It's directly adhering itself to the brain, and it's this very thin layer that again sits directly on top of the brain. And so these guys, as you see it here, the dura mater is the most superficial. The arachnoid and the subarachnoid is the intermediary. It's in the middle, and then the pia mater is the deepest layer. And again, if you look here at this diagram, I don't really care for this diagram. I'm going to skip to this next one here. I like this one better. As you can see here with this diagram, all of the structures that we've just discussed are here. And so if we look, and I'm going to highlight this in red, I think. If we look right in this area and come back up, that area there that I've just highlighted in red is what we respectively known as the dura mater. So that's the dura mater right here. You can see it also comes right along here, comes up, and notice that it also branches down. And so it comes, this brown area right along here is all dura mater. Now, it is that dura mater that helps to create the longitudinal fissure. And you'll notice that there is this term that is looming right over here, known as the fall cerebri. Right there. That is that gap that is found in between the hemispheres of the cerebrum, where the dura mater extends down. So the false cerebri, or the faux cerebri, is that division that goes deep between the hemispheres, the right and the left hemispheres of the cerebrum. So there's the dura mater. Now, directly below the dura mater, you will see this purplish area that I'm going to highlight in orange. That there is the or I should say that there is the arachnoid layer and the subarachnoid layer. Right there. Notice within that arachnoid and subarachnoid layer, you've got a high concentration of blood vessels. That is where the blood vessels are located. Notice, and you're going to see in subsequent pictures, there are no blood vessels. There is no vasculature in the neural tissue itself. We only find vascularization external to the neural tissue, outside of the neural tissue. And there's a reason for that, and that is because basically the cerebral spinal fluid is acting as the blood, if you would, for the central nervous system. The body spends a lot of time and energy protecting that neural tissue. It is a completely sterile environment. It is a completely sterile environment. Cerebral spinal fluid itself is sterile. And the reason for that is because the neural tissue itself is very, very sensitive. To the extent that if blood does mix with that neural tissue, very often what ends up happening is an autoimmune response, where the antibodies within the body actually starts to attack the neural tissue. It doesn't even recognize the neural tissue as being of self, of the same origin. And so the body does spend a great deal of time making sure that there is a separation and a division between that. That cerebral spinal fluid is found within that subarachnoid space. And what will happen is oxygen and nutrients will diffuse across the membranes of the capillaries and the arteries that are found within the external areas of the brain into the cerebral spinal fluid. And other things diffuse across that membrane as well. We talked about nicotine. We know alcohol. Very often vitamins will, hormones will. Drugs will definitely go ahead and cross that, what we refer to as the blood-brain barrier, which is coming up actually it's something that we've already talked about. And so we've been there, done that, so to speak. Now, I do also want to point out, notice there are these nice little extensions coming off of the arachnoid layer. Right? Those nice little extensions right there are what we refer to as the arachnoid villi. What's the job of the arachnoid villi? Well, this is an area where waste products will collect and gather and diffuse back into what we refer to as the superior sagittal sinus. And it's within that superior sagittal sinus where we basically find venous return. We find a collection of veins that are going to be collecting waste products from the cerebral spinal fluid. And that superior sagittal sinus has a direct feed back to the jugular veins, which are going to leave the brain, go down the neck, and empty back into the heart through the superior vena cava. And so, excuse me, structures here that you need to be aware of so far. We know that we have the dura mater. That is already a given. We know we have the arachnoid matter and subsequently the subarachnoid space, the faux cerebellum. We know we also have the arachnoid villi, and those arachnoid villi are going to empty into the superior sagittal sinus. You may also realize, and this is just tying this in, keep in mind, the structure and the organization that we see here is going to be basically, for the most part, the same organization and structure that we see along the spinal cord. And I mention that because there is a space, there is a small gap between the dura mater and the arachnoid layer, which you all have learned as being the subdural space. If you go back to the model of the spinal cord, you will notice that there is that gold or yellow line that kind of runs in between the dura mater and the arachnoid layer. That division is the subdural space, and here we see it within the organization of the meninges as well. And, of course, we can't forget about the subarachnoid space, which, again, is going to contain that cerebral spinal fluid. It's going to contain those blood vessels. And last but not least, you've got this, and it's that bright blue that you're seeing that sits directly on top of the cerebrum. And it's, in fact, going to cover the entire surface of the brain, that being the pia mater. And so you've got everything here that you really need to be able to focus on. I would suggest that you know this diagram frontwards and backwards because, you never know where it may show up. Now, looking internally at some structures, we do have ventricles. Now, the ventricles are basically gaps within the brain. Right? These are spaces or voids that are going to be found within the brain. Within these spaces or these gaps or these voids within the neural tissue, we find a collection of blood vessels that we refer to as the choriid plexus. And that is a horrible job highlighting. It's a little bit better. Which again is this high, dense area of capillaries that is going to be supplying oxygen and nutrients as well as some other things to the ventricle area. Also within the ventricles, we know already that we have ependymal cells, which are going to be producing cerebral spinal fluid. This is, by the way, where we find the blood-brain barrier as well. So we have a high collection of astrocytes that are going to be found within this area. And basically, this structure of having a dense concentration of blood vessels, capillaries, known as the choriid plexus, mixed with the ependymal cells, which are producing and secreting the cerebral spinal fluid, there are 4 of these areas found within the internal structure of the brain. You have 2 lateral ventricles, you have a 3rd ventricle, and a 4th ventricle. And also keep in mind that each of these ventricles are interconnected. And so the lateral ventricle is connected to the 3rd ventricle, the 3rd ventricle is connected to the 4th ventricle, and there is a way for cerebrospinal fluid to leave the 4th ventricles. And we're going to be looking at each of these passageways, if you would, here coming up over the next couple of slides. And so what you're looking at here is basically a rough diagram, if you would, of the ventricle system. You can see the lateral ventricles, which would be right here. Again, these ventricles are going to be associated with each side of the cerebrum. You're going to notice that we have the 3rd ventricle, which is going to be found right here. And what I want to point out to you, and if you look very closely, I think you can see this quite easily, do have these little tunnels, these little pathways that lead out of the lateral ventricles and empty cerebrospinal fluid down into the 3rd. That is what we refer to as the interventricular foramen. Interventricular. In between the ventricles. Foramen is a hole. Interventricular foramen. Right? In between the ventricles, there is an opening or a hole by which the cerebrospinal fluid will empty. And where will it empty into? It'll empty down into that 3rd ventricle. And then you also have the 4th ventricle, which is right here. And you also, again, have a opening or a passageway that leads from the 3rd ventricle down into the 4th ventricle. And that entryway is known as the cerebral aqueduct. And last, we do also have 2 escape routes, if you would, for the cerebrospinal fluid as it is leaving the lateral ventricle, the 4th ventricle. And those are referred to as the lateral aperture and the median aperture. And again, both of these are associated with the 4th ventricle. And basically, the lateral aperture is going to allow cerebrospinal fluid to re-enter circulation around the brain, where the median aperture is going to allow for, I said that backwards. The median aperture allows for circulation of cerebrospinal fluid around the brain, and the lateral aperture is going to allow for cerebrospinal fluid circulation down around the spinal cord. Cerebrospinal fluid itself, we know already, provides protection, provides cushioning. It also, I'm going to add in here, carries and delivers oxygen and nutrients, as well as other things. And it also will remove waste products, right? Things like carbon dioxide and all that kind of good stuff. just an FYI, this is nothing that's going to be on the exam, but researchers estimate about 40% of the cerebral spinal fluid is produced by the subarachnoid space. They estimate that approximately another 30% is produced by the ependymal cells themselves. And then they estimate that the final 30% is also produced by within the choroid plexus itself. Okay, again, that's not on the exam, but that's just more FYI as far as where things are produced. This diagram here, again, just kind of shows you a little bit better as far as where things are leaving and going. And so you can see within here, and we're going to start it right up in here, where the number 1 is. That there is the, let's see if I can get a good, see if this works. What you're looking at there is the lateral. Hold on 1 second. That there is the lateral ventricle. Right in there. And again, within that lateral ventricle, you're going to have the choroid plexus, this collection of capillaries, as well as ependymal cells and cerebral spinal fluid that is being produced. Right here at the anterior portion is going to be the interventricular foramen, right there. And that is going to empty down into the third ventricle, which again is going to be right in this area here. The third ventricle is going to empty down into the fourth ventricle, which is located right here, through the cerebral aqueduct, which I'm going to highlight in red. So the cerebral aqueduct is right there in red. And then from there, and again, keep in mind that the fourth ventricle also has its own escape route, if you would, which is the lateral and the median or the medial apertures. Again, the medial aperture is going to return cerebral spinal fluid flow back around into the brain, and it's the lateral aperture that's going to allow for cerebral spinal fluid directly to flow down around the spinal cord. Here, again, this is just a note on the blood-brain barrier. I don't really have all too much to add to that. Just a couple little statistics. And that is approximately 25% of the oxygen and glucose supply is diverted directly to the brain. Quarter of the oxygen and the glucose that your body is taking in is specifically earmarked for neural function, central nervous system neural function, and that's a big commitment. And hopefully that impresses upon you the importance, if you would, of the brain tissue itself. With that as well, at any given time, about 15% of the body's blood is circulating around the brain. About 15% of the body's blood supply is within the central nervous system at any point in time. Right now, keep in mind, that is specifically referring to the vasculature surrounding that subarachnoid space and going into the ventricles. But again, that's a pretty hefty amount of blood and dedication earmarked specifically for the central nervous system. All right, let's go through here. A lot of this stuff I'm going to be able to move a little bit quicker through because I think you guys have already covered this fairly well. But let's go through and move through the various parts of the brain and make sure that we are okay as far as where things are located. And that's really what I'm going to be focusing on, is where are things located, not so much what they are doing. I think that's stuff that you can look at on your own and learn as well. And so we have the brainstem. We know that the brainstem is composed of three key divisions, if you would. The medulla oblongata, which is the most posterior region of the brainstem. The pons, which is the middle, and the midbrain, which is kind of tucked in to the cerebrum. Each of these guys have their own function. And here you can kind of see where everything is located respectively. The medulla oblongata, I will point this out because this is a pretty critical thing here that it does, is the cardiac center. It does regulate and monitor blood pressure. It does regulate and monitor the force of the contractions of cardiac output within the ventricles. Because it is tied to a circulatory function, it also monitors respiratory function as well. And it's also where we have all the cool reflexes being monitored, non-vital reflexes, coughing, sneezing, swallowing, vomiting, puking, upchucking, ralphing, barfing, blowing chunks, and all those other little colorful terms that you can come up with for vomiting. Again, the pons area, which is kind of right in the middle of the medulla oblongata and the midbrain, influences respiratory control. And so it is working with the medulla oblongata to monitor respiratory function. And it also serves as a passageway, if you would, for the cerebrum and the cerebellum, which we haven't quite talked about yet. But again, you can see directly where that is located, right here in this area. Directly below it is the medulla oblongata, right there. So that kind of puts it into perspective. Another perspective here for you is if you can find the cerebellum, typically you won't have a problem distinguishing the pons and the medulla oblongata. And then you have the midbrain, which is what's going to impact, if you would, reflexes, specifically head and eye movement. So if someone throws a ball at your head and you instinctively kind of take, the hand goes up, the head goes down and away, and you close your eyes. That is all coordinated within the midbrain, and you can't quite see it as well here. This is a bad picture, but you can get the idea, I think. Right here is the midbrain, and then you've got the pons, and followed by the medulla oblongata, which is right there. And so here you can see all three of them. And there's going to be an image coming up here pretty soon where I think I'll be able to point that out a little bit better, possibly, I think, towards the end. Yeah. And we'll label that picture all up for you, all nice and pretty. The diencephalon. Diencephalon is just a fancy word for our next two structures, which is really the thalamus and the hypothalamus. The thalamus is a little bit of a boring structure. It is the hypothalamus where all of the action takes place within the brain. You've heard me refer to it as the party zone of the brain because of what it controls and doesn't control. But before we get to the hypothalamus, let's take a few minutes and look at the thalamus. It is a oval-shaped structure. It does have two lobes to it. One lobe, of course, would be associated with the right hemisphere, one lobe being associated with the left hemisphere of the cerebrum. And basically what it is doing is gathering sensory information. It's gathering information on light. It's gathering information on temperature. It's gathering information on pressure. It's gathering information on touch and pain, which is why there needs to be two lobes, because we know that the right and the left sides of the cerebrum function, in a sense, independently as well as jointly. And so if there was just one thalamus, it would not be able to handle all the incoming processes and sensory information as adequately. And so the right side of the thalamus, associated with the right hemisphere, is actually gathering information from the left side of the body, and the left lobe of the thalamus, associated with the left hemisphere, is really taking in information from the right side of the body. And again, all it is doing, the main function of the thalamus, is to interpret, sort, and respond to sensory information coming in, right? Light, touch, pressure, pain, and temperature. And then there's the hypothalamus. This here is going to be a critical structure for you as you enter into AMP2 and you begin to look at the endocrine system. There's actually four very distinct regions of the hypothalamus for which we do not need to know at this point in time, but know that collectively that hypothalamus controls everything that you see here. It's going to control the autonomic nervous system. It's going to control sensory input from the autonomic nervous system. It's going to control coordination between the endocrine system and the nervous system. It's going to control things such as fear and rage and thirst and hunger and waking cycles and sleeping cycles. It's going to control things such as sexual urges and responses. It's going to control things such as body temperature. Right? All of those things that, and really, if you think about it, it's all kind of interconnected in one odd, weird way or another. Many people who have needs and stresses and pressures right away will turn to food or fluid, as it was, or can't sleep, or sleep too much, or have sexual response issues, or have issues with body temperature. All of these things are all interconnected, and furthermore, they're all really controlled by the hormones associated with the endocrine system, and how we respond to that directly links, as you will find out in AMP2, to the autonomic nervous system. And so the hypothalamus is really, if you think about this, probably the most critically important part of the human body. You will learn about the pituitary gland, and you will learn that it is called the master gland. Bull donkey. Right? It may be a critically important gland, but that gland is taking its directive directly from the hypothalamus. It's taking its directive directly from the hypothalamus. And so that becomes ground zero for so many key functions within the body. Just look at the first three. If you want to know what key functions within the body are, those first three bullets: control over the autonomic nervous system, control over communication between the endocrine system and the nervous system, and how we interpret signals being collected by the autonomic nervous system. You've got T-Rex coming down Main Street, Toms River, and he's coming after you because he wants your Primo's hoagie. You need to be able to effectively gather that input and decide, are you going to run or are you going to defend the honor of your Primo hoagie? The way we respond to outside stimuli and stresses is all found here. It's all coordinated here. And I made mention of the pituitary gland. The hypothalamus is directly connected to the pituitary gland through this nice little stalk, if you would, called the infundibulum. And the infundibulum is that connection that we find between the hypothalamus and the pituitary gland. The hypothalamus very often is creating hormones. It's storing it within the pituitary gland, and then the pituitary gland is taking the controls from the hypothalamus. The cerebrum. You're talking upwards of 83% of the total mass of the brain is found within the cerebrum. And we know that it is the area where memories are formed. All right. In other words, when we create a memory, when we learn things, it is really the cerebrum that is leading the charge on forming those neural connections to be able to store that information. We know that there are 4 distinct areas: the lobes, the frontal, the parietal, the temporal, and the occipital. Don't worry about what they do. I'm going to save that and leave that for the psychology folks to go ahead and go through with you. We're not going to focus on that here, but each of those do have a very specific function and purpose. You know, the frontal lobe is your reasoning, as an example. Right? And so that is all part of that. Now, what I will say is this. We do have some structures that are associated with the cerebrum that we need to be fully aware of. All right? So, in other words, if you look here, you see that we've got what I refer to as the hills. Right there, right there. Let's take that one out. Right there and right there. Those hills are what we refer to as the gyri. All right. You can see that we've got these crevices that are all along the cerebrum. Right? Those crevices, or as I sometimes refer to them as valleys, are what we refer to as the sulci. They're the dips. Now, really, really deep sulci are what we refer to as fissures. And one such example of a fissure is the longitudinal fissure, which we know separates the right and left hemisphere of the cerebrum. The outer layer of the cerebrum is what we refer to as the cerebral cortex, and we know that the cerebral cortex is made up of gray matter. This is also where the gyri and the sulci and the fissures are found within that cerebral cortex. and then just below, just deep to the gray matter, is the white matter. And on this next slide here, you can see exactly that. And so if I can carefully outline for you, see if this works, everything here in orange is representative of the cerebral cortex. Which, of course, then means that this is also where the gray matter is also located. And I think you can see that. You can see that fairly obvious there. And then everything on the inside is the white matter. And I think you can see that clear distinction as well. Now, and I erase that so you can kind of see it a little bit better. Maybe I'll put in some arrows so you can see it as well. So as an example, right there, that there would be, that there would be gray matter, or we can also refer to that as the cerebral cortex. Either way, you are correct. And, of course, right in here, we've got ourselves the white matter. Now, I also want to point out to you that the spaces that are found right here, they are your lateral ventricles, and so you can see the right ventricle here. Actually, no, I lied. That's the left ventricle because it's the back of the head you're looking at, and that there is the right ventricle right there. You can see right here, there's your third ventricle, and then the fourth ventricle is actually right down there. And so you can actually see the ventricle system very clearly in that picture. And the other thing you can really see decently here is this band of white matter coming right through here like that. That band of white matter is known as the corpus callosum, which we know is a collection of axons, which is making up the white matter. Keep in mind that gray matter gets that darker color because it is largely made up of the somatic bodies or the cell bodies of the neurons. And the white matter primarily gets its name because it's largely made up of the axons. And that corpus callosum is that intermediary, is that connector between the right and the left hemispheres. And then we've got the cerebellum. Real quickly, I do want to point out to you, because there are some good things here that I do want to make sure that you have. Again, you can very clearly see here is the pons of the brainstem. Here's the medulla oblongata. And here is the, I lied, that's not the midbrain. Yeah, that is the midbrain. There's the midbrain right there. All right, just kind of pointing that out. And directly behind that is the star of the show, at least for this slide, and that is the cerebellum. And you can see that the cerebellum, like the cerebrum, is made up of white matter and gray matter. That white matter within the cerebellum is referred to as arbor vitae, and the gray matter is the folia. Think of trees. Arbor vitae is a type of tree, and that white matter kind of looks like a tree trunk with branches coming out. And then the foliage, all right, surrounds the branches going around. And if you're looking for a function, again, it's going to control skeletal muscle function. It's going to control location and direction and speed of your limbs. And so how fast your arms are moving, how fast your legs are moving, where your arms are, where your legs are. It does also lend a hand in balance and coordination. And really the last portion here that we need to be concerned with, and I'm going to point out a couple other little structures here as well, but it's the limbic system. And the limbic system here borders or surrounds the diencephalon. And so directly in the middle of the limbic system, we have the thalamus, which you can see very clearly. Right, there's the right lobe. Here's the left lobe of the thalamus. And directly below that is the hypothalamus and the pituitary gland. And the limbic system is primarily made up of two very key structures, the hippocampus and the amygdala. And then it's got some accessory structures that go along with it, primarily the olfactory bulbs, which are involved in smell and scent, and that'll make sense to you here in a couple minutes, as well as the thalamus and the hypothalamus because of hormone control and the sensory input. The limbic system is very much about connecting emotion, which is very often triggered by the sense of smell or the sight of something, and a memory. And so the hippocampus is involved with taking memories that are short-term and converting them into a long-term memory that is then stored within the cerebrum. The amygdala takes a sense. It takes something along the lines of smell or sight or vision or texture, and it links it to this long-term memory. So you walk in a room and you smell a cigar, and you think of your great-great-grandfather, Fred, right? Because that smell of the cigar is something that you associate with him. Or you walk by a bakery and you smell an apple pie baking, and all of a sudden you remember holidays with your great-aunt Tilly, because great-aunt Tilly would always bake apple pies during the holidays. That's what the limbic system is doing. It's that amygdala, which is the end bulb, really, of the limbic system. It takes that emotion and it links it to a memory that was formed by the hippocampus, and again is stored within the cerebrum. And here is the brain. And so what you really need to be able to do is identify and label all of these various structures that we have talked about. Being able to point at them and say, all right, that structure right there is it's part of the cerebrum. It's also gray matter. It's made of gray matter. It's referred to as the cerebral cortex. And within the cerebral cortex, we have gyri, which are the hills, and we've got sulci, which are the dips or the valleys. And I think you can see all of that stuff very clearly. You can see very clearly the white matter that is located within the cerebrum. And the white matter is white because it's primarily made up of axons. And then we go into the various structures, such as that right there, which is the corpus callosum. And directly below the corpus callosum, we have the lateral ventricles. And right here in this area, right there, we have the thalamus. Right above the thalamus, you have the third ventricle. Right below, right in this area, you've got yourself the hypothalamus. Right there would be the midbrain. That structure right there is the pons. That structure right there. The hypothalamus. Of course, here we've got the cerebellum. Right here. You got yourself the fourth ventricle. All right, and so all of this stuff begins to play itself out. You should be able to identify all of these regions. One of the areas that I did not discuss with you is actually an area that is right here, and that would be the corpora quadriminal, which again is made up of the superior and inferior colliculi, which again helps to coordinate hearing and vision specifically. I think you can see very clearly here also that this black space that is surrounding it right there, that there becomes the subarachnoid space, which is where the cerebral spinal fluid is flowing around. So this is a really good image here for you to be able to, and it's in the PowerPoint. I would suggest taking this and making sure that you can label everything that is on there. And one last little slide. You should be able to identify everything that is on here as well. This is the big test. You know, if you can label all the parts of this sheep brain, then you know you are ready for the lab practical as well. And so this here, of course, is the corpus callosum right there with the lateral ventricle being found right there. This here is white matter of the cerebrum. Third ventricle. This here is folia or gray matter of the cerebellum. This here would be the arbor vitae of the white matter of the cerebellum. Of course, this right here is the fourth ventricle. And then we've got, respectively, we've got the medulla oblongata right here. You've got the pons right there. You've got the midbrain right there. And collectively, that's going to make up the brainstem. One of the structures we did not talk about but do not worry about is the mammary body. Here is the hypothalamus. The optic chiasm. And last but not least, right here is the thalamus. And I'm going to add in a structure or two Right there is the pineal gland, and right there, that's the corpora quadriminal, right there. And so as long as you should be able to identify all of those reasons, you should be just fine for the lab practical, as well as the lecture exam itself. And with that, I know I am running way over an hour, about an hour and 10 minutes. I apologize for that. But we are done. You have survived. Congratulations. Boom! Confetti's going off, fireworks launching over your head. If you have any questions, please, 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 please let me know. It's been fun. And for those of you that I will see in A&P2, rest up, because we'll be having a lot more fun there.